Now we're going to show you a couple of different circulatory routes, what we call a typical circulatory route, and then a portal circulatory route. First of all, the typical circulatory route has vessels leaving the heart. They break out into smaller and smaller arteries, so we have a number of arteries that keep branching and branching. And our smallest arteries right here are called arterioles. Arterioles then branch into our smallest vessels, our capillaries. Capillaries, your smallest vessels, and that's where you have the exchange of oxygen and carbon dioxide, where nutrients and hormones will be delivered to your tissues, and waste products will be picked up and removed from your tissues. They're in the capillaries. Capillaries then come together as venules. These smaller vessels here, and venules come together to make larger and larger veins on the way back to the heart. So we came from the heart over here, out through arteries into smaller and smaller arteries. Arterioles then lead into capillaries. Capillaries come together to make venules. Venules then come together to make veins, and then larger and larger and larger veins as they come together with one another on their way back to the heart. So this is a typical circulatory route. Most of the body is supplied by this kind of typical circulatory route. Most of your skeletal muscles, your bones, your skin, different regions of the body are supplied by a normal or typical circulatory route. Next we're going to talk about portal circulatory routes. So here we see what we call a portal circulatory route. We start again with the arteries, then the arterioles, and then here's a capillary bed. And the capillaries here convene not into a vein, but in orange here on the board, we have a set of vessels that come together and then branch out again. These are called portal vessels. Portal vessels. Portal vessels then lead into the second capillary bed. Not room to put it all in here. Those come together in venules. And then finally veins. That will head back toward the heart again. So we have two capillary beds in our circulatory pathway between the arteries and the veins. Examples of portal circulatory pathways in the human body include the hepatic portal system, where blood from the stomach, intestine, pancreas, and spleen are routed, blood from those organs will be routed into the hepatic portal vein and go to the liver. In the liver, it breaks out into small passages again. We call them sinusoids instead of capillaries typically, but they're the equivalent of capillaries in terms of this model here. And then they reconvene within the liver as hepatic veins that leave the liver from there. So two sets of capillaries in the route. Another location where we have the portal system in the body is between the hypothalamus of the brain and the pituitary, so that hormones from the hypothalamus will be gathered up into the portal vessel and delivered directly to the pituitary gland. There. So we'll describe both of those things next. Now this is kind of a lousy picture we have here, but the liver looks all right up there. This would be the stomach region right here, the spleen over there on the side, the pancreas, this structure right here, and then all this small intestine in here, and then the large intestine running around here. So we have blood vessels that come from all of those locations that feed into the liver. Right up in here is where it would be entering kind of back underneath the front lip of that liver there. But anyway, this large hepatic portal vein comes through all these membranes, mesenteries behind the stomach right there, but has branches that reach out across the stomach, a branch that reaches out to the spleen, branches that go into the pancreas, and then all throughout down here in the intestines. So blood from all of these organs, the spleen, the pancreas, the stomach, the intestines, all that region right there, after the blood comes to them and circulates through the capillaries of those organs, that blood is collected into the hepatic portal system. And all of those blood vessels, hepatic portal vessels, collect into this main hepatic portal vein right here. In the liver, that branches out alongside vessels of the hepatic artery. So the hepatic artery branches and the hepatic portal vein branches all end up going to the same locations within the liver, supplying those small liver sinusoids, those small capillary-like spaces inside the liver. And so anyway, that's your hepatic portal vein arrangement. In this picture that we have on the board here, this region across here would represent the hypothalamus. Hypothalamic region of the brain, or hypothalamus. And it has an extension down here called the posterior pituitary, and then another part here called the anterior pituitary, or adenohypophysis. So there's a circulatory route that supplies the hypothalamus here, the blood supply that comes into the hypothalamus, branches out into its capillaries, and then that's going to collect into a portal vessel, small portal vessel, where these vessels come together and supply one vessel that extends in toward the pituitary, and in the pituitary that branches out again. I guess I should make that branch out in orange here, supplying all of the pituitary capillaries, and then that blood collects into small venules that will leave the pituitary. So this we're just going to call this the hypothalamus / pituitary portal system. So blood is supplied here to the hypothalamus. There are hormones produced in the hypothalamus that help control activities of the anterior pituitary. So that's circulation there. They pick up those hormones and deliver it directly in a portal system to the tissues of the anterior pituitary here. And then that blood returns then through veins to leave the pituitary. So arteries, arterioles, capillaries, portal system, and the capillaries again, and then the veins, venules and veins that leave. A normal portal system. The human heart exists within what we call the pericardial cavity, and there are membranes that attach to the heart, kind of wrapping around from the top up here. There's one membrane that adheres directly to the surface of the heart, and I'm trying to draw that now, and then it reflects back out around from the top of the heart to make this heart chamber, the pericardial cavity, like this. So that layer immediately against the heart muscle in here is referred to as a visceral pericardium. And that outer wall layer of membrane out here is called a parietal pericardium. And between the two, there's not really a cavity, a space like filled with air or something like that, or even very much fluid, just a very thin layer of what we call a serous fluid, a watery fluid in there. It's a little bit slippery, but anyway, that's our pericardial cavity. So that parietal pericardium and that visceral pericardium come together near here, the top of the heart, where all the vessels are attached. This is considered the base of the heart. Up at this end, the base of the heart is at the top of the heart, and the other end down here is referred to as the apex of the heart. The apex of the heart. Most of what we see here is ventricular muscle on the front, and then to either side we have atrial chambers. So there's an atrium and there's an atrium. At the edges of the atrium, there's a little bit of a loose edge kind of extending out over the top of the ventricle here. And that edge, that flap, is called an auricle. Auricle. It's just the flap of the atrium that kind of extends out over the surface of the ventricle in here. So all of this is ventricular muscle. That's all ventricle in there. Internally, we'll see that it's divided into a left ventricle and right ventricle. Up here we have the right atrium and left atrium above. So the heart is in a chamber called the pericardial cavity. On the heart surface is the visceral pericardium membrane, and out on the others, out to the outside wall of that pericardial cavity is our parietal pericardium. Next, we're going to look at the thickness of the heart wall itself. All right, if we look at the thickness of the heart wall between the inner chamber and the outside, there are layers to the material in there we call endocardium, myocardium, and epicardium. The thickness of the wall on the left ventricle is really thick, a little less thick on the right ventricle, but in the atrial chambers it's much thinner, and what I've drawn here is pretty much what you'd see on the wall of an atrial chamber. The thickness of the wall right here is what we're looking at. Now, on the inside, there's going to be an endothelium. Same name we give to this simple squamous epithelium in the blood vessels. It's also in the heart. There's going to be a layer of connective tissue in there, and those two things make up our endocardium. So we have our endocardium. Then we have a thick zone that's made up of muscle. Now in this muscle zone, there can be quite a bit of connective tissue. There might be blood vessels within this muscle, and there could be even some adipose scattered in there among this muscle, but mostly it's cardiac muscle with some connective tissue. And this is the myocardium. Myo always refers to muscle, so that's the muscle layer of the heart. And then to the outside we have another connective tissue layer, and we have a mesothelium. A mesothelium layer, and that all makes up the epicardium. Now this epicardium portion right here is essentially the same thing as the visceral pericardium of the heart. So visceral pericardium, epicardium, essentially the same thing. I want you to know how to draw the heart, and I want you to do it this way. This is a simple way of learning the heart anatomy and how the heart works. Start with a shape something like this. You have two atrial chambers and two ventricular chambers down below. Start kind of in the middle here between these 2 points and draw a line up like that, curving that away. And then another one coming up here, and this looks like a tree trunk that branches there at the top. from this point right here all the way to the bottom. That will divide the two ventricular chambers. So now we have a right atrium and a left atrium, a right ventricle and a left ventricle chamber of the heart. This is the pulmonary artery right here, and behind that pulmonary artery, we want to draw in the aorta. So it goes in behind that and up and over the top of it. I want to close in my heart right there, so everything's sealed up. So now we have the two atrial chambers, two ventricular chambers, a pulmonary artery, and the aorta. Blood coming to the heart is going to enter over here in the right atrium, and these enter on the back side of the heart. So there's a superior vena cava coming down from the top up here, and inferior vena cava coming in here. So that's the return of all of the systemic blood, the blood that's been to all of the tissues of your head, your legs, and so forth, all coming back to the heart here on the left side. Over on the other side, this blood comes in from the lungs. So we have a couple of pulmonary veins over here. And there would be some pulmonary veins coming in from the other side also. I'm going to draw two circles to represent that. They would be way on the back side of the heart over there. But anyway, that's pulmonary veins, where blood is coming in from the lungs. So those are vessels and chambers we already have on there. Now we're going to put in our valves. I'm going to put my valves in a different color just to make them show up a little differently here. I draw the valves as little arc shapes in here, like this. These valves between the atrial chambers and the ventricular chambers are going to be called atrioventricular valves. And at the base of the pulmonary artery and the aorta, they're called semilunar valves. So we have a right atrioventricular valve and a left atrioventricular valve. We have a pulmonary semilunar valve and we have an aortic semilunar valve. We're going to go ahead and go through this and label all the parts now. Starting with the blood coming in from the systemic circulation. Here's superior vena cava. Coming down. And then the inferior. Down here. And they both bring blood into the right atrium. The right atrial chamber, then that blood is going to be pumped through this valve right here. And we're going to label that. Let's see, I'll put a little arrow to it right here. This is the right atrioventricular valve. Right atrioventricular valve, and that blood then moves into the right ventricle. Blood from the right ventricle will be pushed out of the heart through this pulmonary artery right here, and it will pass through the pulmonary semilunar valve. And there's not a good way to label that here. Let's draw a line up through there and label that. Pulmonary semilunar valve there. So that blood goes through the pulmonary artery. To the lungs, comes back to the heart through pulmonary veins, and these are pulmonary veins. As are these over here on this side. So anyway, the blood's coming in to our left atrium. passes from the left atrium to the left ventricle through this valve right here. The left atrioventricular valve. That blood then enters into our left ventricle. From the left ventricle, it's being pushed up through the aortic semilunar valve. And out the aorta. So that drawing is a drawing of our vessels, valves, and chambers. The vessels, valves, and chambers. So, 4 chambers, right atrium, right ventricle, left ventricle. The vessels, superior and inferior vena cava, bringing blood in. Then that blood is sent through pulmonary artery to the lungs and back. Comes through pulmonary veins back to the heart. Goes from the atrium to the ventricle and then out the aorta. The valves, atrioventricular valves between atrium and ventricle on the right side, and then another left atrioventricular valve over here on the left side. Then the semilunar valves keep the blood from coming back down into the ventricles from the pulmonary and the aorta. The pulmonary semilunar valve here and the aortic semilunar valve there. Next, we're going to look at the path of blood flow and talk about the function of valves again. Okay, we're going to start by drawing our heart again. Our basic shape, and then our pulmonary artery. Then divide the ventricles. Then put the aorta behind the pulmonary, coming up and over the top of it up here, close in my heart right there, put in my superior vena cava, and then my inferior vena cava, my pulmonary veins coming in from the left and on the right over here, put in my valves, my right atrioventricular valve, my pulmonary semilunar, aortic semilunar, and then my left atrioventricular valve. So there's all the parts put back in. Now to talk about blood flow through the heart. All the blood that has been to the top of the head, all the way to the bottom of the feet, all those regions are going to come back into the heart. This is what we call systemic blood, coming from the inferior vena cava and superior vena cava into the heart here in the right atrium. From the right atrium, this right atrium will contract and push the blood down through that valve into the ventricle. When the ventricle contracts, after the atria contract, they will push the blood back out of the ventricle, but this valve will keep it from going back into the atrial chamber. Instead, it will be pushed through the semilunar valve and out the pulmonary artery up here. So that's deoxygenated blood, and we use the blue color to indicate deoxygenated blood. So, receives systemic blood from the vena cavas into the body. Oxygen has been removed in all the tissues out there. So then pumps to the lungs to get our oxygen. After it's been to the lungs, it comes back through these pulmonary veins, empties into the left atrial chamber of the heart. From the left atrium, it's pushed down into the left ventricle through the left atrioventricular valve. From there, it's going to be pushed back up from the ventricle into the aorta. And the aorta will then carry it to all the systemic circulation. Everything that the vena cavas here will retrieve the blood from are supplied by the aorta. So this is systemic circulation from the aorta to all the body, and then recollecting eventually over here from the vena cava. pulmonary circulation. This blue line here in the pulmonary artery indicates deoxygenated blood that will go to the lungs and then get oxygen. When it comes back, it's going to have oxygen and have the red arrows here. So this is the pulmonary circulatory route, from the pulmonary artery to the lungs to the pulmonary veins. So we have a systemic route from the aorta to the body to the vena cava, and a pulmonary route from the pulmonary artery to the lung to the pulmonary vein. Now, when the heart beats, when you get contraction of the heart chambers, both atrial chambers will contract at the same time, opening and keeping these atrial ventricular valves open on both sides, allowing that blood to move down into the ventricles. Then the ventricles contract after the atrial chambers have already contracted, so they're starting to relax. The ventricles start to contract. It closes the atrial ventricular valve so we don't get movement of blood in the wrong direction. We keep things going the right way. It opens up these semilunar valves at the base of the aorta and the pulmonary artery, and blood is pushed up into both of those vessels, both the pulmonary artery and the aorta. So we see the blood pushed up here, and after the ventricle is finished contracting, it starts to relax. We don't want that blood to come back down, so these valves close to keep the blood from moving backwards from the major arteries, the pulmonary artery and the aorta, back into the ventricles. Another consideration when working with the heart and anatomy is the electrical conduction system, how the electrical heartbeat, that action potential spread occurs in the heart. Any tissue in the heart will have what's called autorhythmicity. That means it has the ability to generate an action potential that will cause a contraction of that muscle. So if you had ventricular muscle or any other kind of muscle, took it out and put it in a dish with normal saline for a while, you would see it undergo normal, regular contractions on a predictable kind of cycle. But there's one place in the back of your right atrial chamber back here that will undergo that normal depolarization, that autorhythmicity, at a faster rate than other muscle tissue in the heart. And that is often referred to as the pacemaker of the heart, because once it starts here, it spreads to other parts of the heart. So it sets up the overall pace for everything, and its technical name is the sinoatrial node. Sinoatrial node right there. Once it undergoes its depolarization, it forms an action potential. That action potential will spread through both atrial chambers. Both the right side and the left side will get that spread of the action potential, so that both of the atrial chambers contract basically at the same time. Even though it starts here in the right atrial chamber, they both contract pretty much at the same time. Where this green line is across here, separating the atria and ventricles, where those valves are, there's actually a connective tissue partition that keeps the muscle of the atrium from coming in contact with the muscle of the ventricle. So there's kind of an insulating layer there so that this action potential up here is not spreading automatically to ventricular muscle. But there is a location in the heart back in here that will receive that signal and then send it down. I didn't split my ventricle, did I? And send it down the interventricular septum. From here it will divide out and go to both sides of the heart, both the right and left ventricle. And then there are extensions of that pathway that lead up into the muscle of the ventricle on either side. This node right here is the atrioventricular node. The atrioventricular node, there's a little opening, I guess, in that connective tissue that allows that signal to pass through along what's called the bundle of His. So it travels down the bundle, and then it goes into what are called right and left bundle branches. There's the left and there's the right. Left and right bundle branches. And then those radiating fibers that conduct the signal out into the muscle of the ventricle are called Purkinje fibers. Purkinje fibers. So the spread of the electrical conduction, that action potential, goes through both atrial chambers. It reaches the atrioventricular node that sends it down the bundle of His. And then we have the split to the bundle branches, left and right, and then out through the Purkinje fibers to the muscle of the ventricles, and the ventricles contract. So there's a delay between the atrial contraction and the ventricular contraction. While we're on the subject of the electrical conduction in the heart, the action potential traveling from atria to ventricle and so on, it's a good time to look at the electrocardiogram. And the electrocardiogram, the typical trace, I've kind of roughly drawn out here, consists of what's called a P wave. That represents the depolarization of the atria that results from that sinoatrial node, that pacemaker depolarization. That's where things start. It then travels from there to depolarize all the muscle of the atrial chambers. And that's what we see in the P wave, is that depolarization of the muscle in the atrial chambers. Eventually it will reach the atrioventricular node and send the signal out to the ventricles. And this is what we call a QRS complex. It's the Q, that's the R, that's the S. So we call it QRS complex. And that's the depolarization of all the ventricular muscle, all the ventricle muscle contracting there. So there's a lot more muscle in the ventricles, so there's a whole lot more exaggerated disturbance in the electrical situation or state in the heart for that. Following the QRS complex, there's a brief period of time before we have kind of a return to normal, where all of the ions that had to move across membranes in the QRS complex go back to their normal state. So it's a repolarization of the membrane, getting things back to normal, ready for the next contraction, and that is the T wave. So this is ventricular depolarization, this is ventricular repolarization here. Why do we not see a repolarization of the atrial chambers? Well, it would be rather small. If we could see it, it would be a small blip in here, and it kind of would be, it's covered up in this QRS complex. Its recovery, its repolarization happens in this time frame. So there's a depolarization here, repolarization there, but it's covered up in QRS complex. And then we get the T wave, the repolarization of ventricular muscle out here. It's a little more profound, actually produces a significantly larger wave than the P wave depolarization that we started with. So this is the electrocardiogram, and that's how it relates to what we just talked about with the sinoatrial node and the atrioventricular node and the traveling of down the bundle of His, the bundle branches, and through Purkinje fibers. Now a little about arteries and veins. First of all, we're going to talk about the basic layers of an artery. So this is an artery. This would be a small artery, not a very large artery. The vessel would be comparable to maybe a vessel going to your finger or something of that sort. Just a very small vessel carrying arterial blood, oxygenated blood. It consists of 3 layers, this artery, and most medium to small arteries are like this. There's a layer to the outside that has connective tissue we call the tunica adventitia. An adventitia is usually a connective tissue layer that just surrounds an organ and makes contact with surrounding connective tissues from other organs and so forth. So anyway, so we have an outer adventitial layer. Next we have a layer of smooth muscle. It's completely in smooth muscle. It's called tunica media. The middle layer, the tunica media. And then finally we have the third layer, and I've drawn it a little thicker than it should be here, but anyway, this is called the tunica intima. Tunica intima, that inner layer. Sometimes it's called the tunica interna, and sometimes the tunica adventitia might be referred to as a tunica externa on a vessel. There are different layers to the tunica intima in the artery. There is a layer of, the pink line right there represents endothelium. That's simple squamous epithelium in there. Underneath of that, there's a little bit of connective tissue, a thin layer of connective tissue. And then this orange line represents the internal elastic membrane. Internal elastic membrane. So those three parts right there make up that tunica intima. The tunica media was made of smooth muscle. Smooth muscle and the tunica adventitia, that's all connective tissue out there. There may be a little bit of elastic fiber out there in that tunica adventitia. It's very common for there to be an elastic component on the outside of any blood vessel like that. So anyway, that's a cross section of an artery. Next we're going to draw a vein that would be a comparable vein. It's common for in something like your fingers or in your hand somewhere or any other organ that there's an artery going out and a returning vein, returning the blood the opposite direction, and they sit side by side typically or very commonly in the tissue. So you'll find an artery-vein pair. So let's draw the vein that would be comparable to that. So a few things you're going to notice about the vein. First of all, compared to the artery over here, it has a much larger cross-sectional area. The area within that cross-section of the vein is much larger. I've drawn it kind of odd shaped, not circular. So it's very common for the veins to not maintain a nice circular shape. They don't have as much of a layer of smooth muscle around them. So let's look at some other features here. First of all, they do have that tunica adventitia. That is connective tissue, and it's much thicker in a vein than it is in the comparable artery. In a vein, the tunica adventitia is going to be a thicker layer than the tunica media layer, that smooth muscle layer. So next is the tunica media. The smooth muscle layer. And again, it's much thinner here than it is in a comparable artery, even though the overall cross-sectional area is greater. On the inside, we have this tunica intima. And there's no internal elastic membrane in that. No internal elastic membrane. So it just has the endothelium and a little bit of connective tissue that lies to the inside. Did I say that right? It has the endothelium and a little bit of connective tissue that lies inside of the tunica media, the smooth muscle layer, and fairly thin smooth muscle layer compared to the tunica adventitia and compared to what you would see in an artery. Tunica adventitia here in the vein is thicker than what you'd see in the artery, and it is thicker than what the smooth muscle layer is in the vein. So next we're going to make a table of those comparisons between the artery and vein. So here's my simple table of comparison between a medium artery and medium vein. Arteries have a puckered lining, the internal shape. Because the smooth muscle kind of shrinks up the inner lining, it's thrown into those folds that we see to make a puckered lining in a medium artery, while a vein would have a smooth lining to it. It wouldn't be thrown into those folds. The medium artery has an internal elastic membrane. No internal elastic membrane in a medium vein. Arteries have a smaller luminal area. The cross-sectional area, total cross-sectional area of a medium artery is smaller than what you would see in a vein. A vein has a larger luminal area. Artery has a thicker tunica media, more smooth muscle layer. More thicker smooth muscle layer, that tunica media, compared to a vein. The vein has a thinner tunica media. The tunica adventitia in an artery is relatively thin compared to the tunica media and compared to what you'd find in a vein. So a vein has a thicker tunica adventitia, thicker than the tunica media, thicker than what you find in an artery. So those are the comparisons, important comparisons between an artery and a vein when you see them in cross-section in a tissue slide. A few other odds and ends about arteries and veins. Our largest arteries are often referred to as elastic arteries. Elastic arteries have a much thicker tunica media than even a medium artery. Usually we think of our aorta when we think about elastic arteries, but some of the major vessels that stem off of our aorta would also be considered elastic arteries with a very thick tunica media, and in that tunica media there's a lot of elastic material in that tunica media. I'll put with elastic material. And that is not exactly like the elastic fibers we see elsewhere in the body. It's produced actually by the smooth muscle fibers of the tunica media, this elastic material that you see in there. These vessels do not constrict and dilate according to the need of the tissues in the locality like medium arteries, or what we sometimes refer to as muscular arteries, would. Elastic arteries expand due to the pressure of the heartbeat. When the ventricles contract and push that blood up into those vessels, they are put under tension, and that tension is from elastic materials kept on the vessel as that pressure goes down and that blood is dispersed out to all of the smaller arteries around. So elastic arteries are a little bit different from medium arteries in the presence of this thick elastic layer and in the way that they behave. More like a garden hose, and they don't constrict and dilate so much. Some other notes here. The smaller venules and the capillaries do not have any extra layers beyond the endothelium. So capillaries and venules, let's say endothelium only. The smallest arteries with smooth muscle on them, called arterioles, lack the internal elastic membrane. So they look a little bit different from a medium artery, and just not having that internal elastic membrane. They're a much smaller version of an artery otherwise. They have the smooth muscle layers, but there may only be 2, 3, 4 layers of smooth muscle around a very tiny puckered lining in an arteriole. So that's a little bit of the variation we see from the normal artery vein construction. Elastic arteries, the largest arteries, you don't see as much constriction and dilation of the thing. It's just stretched by pressure, and the elastic material keeps the pressure on it in there. So they're a little bit different. Capillaries and venules are only just a layer of endothelium. There's not usually much connective tissue at all as far as an encasing layer around those vessels. No other material making up the wall of those things. Remember, that's where you have the exchange of oxygen, carbon dioxide, and all kinds of other materials between the tissues and the blood. So we don't want any barrier in there. That's where we have the exchange. Arterioles, like very tiny arteries, but they don't have the elastic membrane present.